everyone. So in this video we'll be learning about microbiology and many other concepts relating to this microbiology and their significance in understanding this microbiology. So in this video we'll be focusing on the invisible world, the birth of microbiology, the science of taxonomy, the role of genetics in modern taxonomy, the binomial nomenclature, and types of microorganisms. Okay, so before we get into those concepts that I just mentioned, we have to look at what is microbiology as a field of study. Okay, so microbiology is the study of microorganisms and all the other living organisms that have microscopic properties or characteristics and maybe functions. Okay, so an example would be the parasitic helminths. Okay, so parasitic helminths are technically macroscopic, but they are included in the field of microbiology because they have microscopic larvae and eggs. Now let's look at the invisible world. So we'll look at what our ancestors knew regarding microbiology or microorganisms. Okay, so this is usually the concept regarding fermented foods and beverages. Okay, so people across the world have enjoyed fermented foods and beverages like wine, bread, cheese, yogurt, pickled vegetables. Our ancestors were making fermented beverage from rice, honey, and fruit as early as 7000 BC. Okay, so one good of example of fermented beverage here in South Africa, it's umqombothi. So production of these foods and beverages, they require microbial fermentation. This is a process that uses bacteria, mold, or yeast to convert sugars to alcohol, gases, and organic acids. So the beer that's made here in South Africa, it's actually made using the yeast, probably the yeast Saccharomyces cerevisiae. Well, I meant to say Saccharomyces cerevisiae as being the microorganism, not actually the yeast, but being the microorganism present in the yeast that's used to make beverage or that fermented beer. The Iceman Treatise. So our ancestors believed that illness was punishment for angering the gods or was simply as a result of fate. They attempted to treat illnesses and infections. One good example of this is Ozzy the Iceman. So some researchers think that 5,300 years ago, the Iceman used natural laxatives and antibiotics. Example, the woody fruit of the Piptoporus betulinus fungus. Piptoporus betulinus fungus, which has both laxative and antibiotic properties. And our ancestors also used herbs as one of the medicinal plants to treat illness and infections. So now the early notions of disease, contagion, and containment. So ancestors believed that diseases were caused by things they could not see. A great example is that the Bible refers to the practice of quarantining people with leprosy and other diseases, suggesting that people understood and knew that diseases could be communicable. Alright. So communicable means that these diseases can be transmissible from one person to another. Now we're going to look at the Greek physician Hippocrates. This man dismissed the idea that disease was caused by natural and not supernatural forces. He then posited that diseases had natural causes from within patients or their environment. This is interesting. The Greek philosopher Thucydides, this one made an observation about the Athenian plague that killed 1/3 of the Athenian population. This was to show the early understanding of immunity. Alright. Then Marcus Terentius Varro, this guy proposed that things we cannot see can cause diseases, and preventative or precautionary measures should be taken, as these microorganisms float in the air. They can enter the body via pores. Now let's look at the birth of microbiology. What brought about this study of microorganisms? So a Dutch law merchant, Anthony van Leeuwenhoek, was the first person to develop a lens powerful enough to view microbes. He built a simple microscope, the most basic microscope, even though the probable developer of the simple microscope is said to be Zacharias Janssen. Okay, so Leeuwenhoek was able to observe the single-celled organisms, which he described as animalcules or wee. These are little beasties swimming in a drop of rainwater. And presently we know that animalcules and we that he was referring to, they're actually bacteria and protists. Okay, so between the ages of 1857 and 1914, the two famous microbiologists, Louis Pasteur and Robert Koch, they were active in advancing our understanding of this unseen world, of this world of microorganisms. This was during the golden age of microbiology. Now let's look at Louis Pasteur and his contribution to the birth of microbiology. So Louis Pasteur was a French chemist, and most of us know this man or this scientist from his experiments for refuting the spontaneous generation and also fermentation. Alright, but he also invented pasteurization, which is a process used to kill microorganisms responsible for spoilage, and developed vaccines for the treatment of diseases including rabies in humans and animals. Now, Robert Koch, this man was a German physician. He was the first one to demonstrate the connection between a single isolated microbe and a human disease. Okay, he made postulates. They are known as Koch's postulates. And he was the first person to discover the microorganism Bacillus anthracis, which causes anthrax. Now, the science of taxonomy. So, taxonomy is the classification, description, identification, and naming of living organisms. You can pause this video and note it down. Taxonomy is the classification, description, identification, and naming of living organisms. So the Swedish botanist, zoologist, and physician, Carolus Linnaeus, this man developed a new system for categorizing plants and animals. He divided the natural world into three kingdoms. We know them: the animal, the plant, and the mineral kingdoms. Right, so the names of the Linnaeus original taxonomy were kingdom, class, order, family, genus, and species. Alright. So the trees of life have evolved throughout the years, the phylogenies. All right. So in the 1800s, there was a growing interest in developing taxonomies that looked at the evolutionary relationships of all the different species of organisms on Earth. This was actually via the phylogenetic tree, right? So in the early phylogenetic trees, the relatedness, how these organisms relate to each other, the relatedness of organisms was shown by their visible similarities, such as the presence or the absence of hair or number of limbs. While nowadays, phylogenetic analysis, they include genetic, biochemical, embryological comparisons. They are way too complex and advanced. So Linnaeus tree of life contained two main branches for all living things. There was animal and plant kingdom. Ernest Hegel later proposed another kingdom, which was the fourth kingdom, the kingdom Monera, for unicellular organisms whose cells lack nuclei like bacteria. In 1969, an American ecologist, Robert Whittaker, this man proposed adding a new kingdom in his tree of life. This was the kingdom fungi. Okay, always remember this. Robert Whittaker proposed adding the kingdom fungi, while Ernest Haeckel proposed adding the kingdom Monera and the kingdom Protista. The kingdom Protista was for unicellular organisms, and kingdom Monera was for unicellular organisms whose cells lack nuclei like bacteria. Okay, so back to Whittaker. So Whittaker, in history, there was a level of categorization above kingdom level, empire, or super kingdom level. This was to distinguish between organisms that have a membrane bound nuclei in their cells, which are eukaryotes, and those that do not have a membrane bound nuclei, which are prokaryotes. Now let's look at the role of genetics in modern taxonomy. You still remember what taxonomy is? All right, so genetic methods, they allow us to compare all the living organisms without relying on their observable characteristics that can be subjective. All right. So modern taxonomy, it relies heavily on comparing the nucleic acids, your DNA, your RNA, or proteins from different organisms. So the more similar the two nucleic acids and proteins are between the two organisms, the more closely related they are considered to be. So there are contributions made by microbiologists. The first microbiologist is Carl Woese, and the other one is George Fox. Okay, so Carl Woese and George Fox, they created a genetics-based tree of life, which was based on the similarities and differences they observed in the small subunit of the ribosomal RNA of different organisms. And they discovered that a certain type of bacteria, the archaea bacteria, were significantly different from other bacteria and eukaryotes in terms of the sequence of the small subunit of that RNA. So the phylogenetic tree that is made or proposed by Carl Woese and George Fox, it led to the three-domain system, which is the bacteria, the archaea, and eukarya. And the bacteria and archaea domains contains all prokaryotic organisms, and the domain eukarya contains all eukaryotic organisms, and it is large among the three domains. Okay, this gives it more reasons why it's large among the other three domains, because we know eukaryotic organisms have compartments, and also with their DNA is long and linear. Okay, so now the naming of microorganisms. So for naming microorganisms, we use the binomial nomenclature, which was developed by Carolus Linnaeus. It's a taxonomy developed by Carolus Linnaeus. It's actually a two-word naming system for identifying organisms by their genus and species. A great example is the Homo sapiens, and there is a way in which this binomial nomenclature is used. Now we have Badges manuals. Okay, these are the manuals that are used for identifying and classifying different prokaryotes. Okay, we have Badges Manual of Determinative Bacteriology and Badges Manual of Systematic Bacteriology. This had to be put in place because many bacteria look identical. Okay, even though if they can be rod-shaped or spherical shaped or comma shaped or spiral, they might look identical, but they might be from different species. Okay, so methods based on non-visual characteristics must be used to identify them, and also be able to distinguish different types of bacterial species. For example, biochemical tests can be used to identify chemicals unique to certain species. Also, serological tests can be used to identify specific antibodies that will react against proteins found in certain species. Okay, now to the last point that we're going to look at in this video is the types of microorganisms. Okay, so microorganisms are found in each of the three domains. You remember them, the domain from Carl Woog's phylogenetic tree, the archaea domain, the bacteria domain, and the eukarya domain. Now let's look at the prokaryotic microorganisms. These are these microorganisms that fall under the two domains, the archaea domain and the bacteria domain. So under prokaryotic microorganisms, we have bacteria. Bacteria are found nearly in every habitat on Earth, including within and on humans. Okay, they are ubiquitous. They are found almost everywhere. Okay, so some bacteria are pathogenic, meaning they can cause disease. I know all of us know that, but most of them are harmless or helpful. We usually use them for fermentation. I'm just kidding. There are many other applications where we can use microorganisms or bacteria, to be specific. So the prokaryotic organisms, most of them have peptidoglycan on their cell walls. This is very important. It's very important to know this. Most bacteria, which is under prokaryotic organisms, they have peptidoglycan in their cell walls. And we have common shapes for bacteria, which are spherical, being coccus, rod-shaped, bacillus, and curved, which can be spirillum and spirochete on vibrio. Although I prefer to say vibrio is comma-shaped, but these ones are the most common one. Hence we say curved, they are spirillum, spirochete, or vibrio. Alright. And we also have photosynthetic bacteria such as the cyanobacteria. the N-oxygenic green sulfur bacteria and the green non-sulfur bacteria. Okay, those are this photosynthetic bacteria. We will look into more detail of these kinds of bacteria in more sections, more videos to come. Okay, so there are bacteria that also use energy derived from sunlight and fix carbon dioxide for growth. And we have non-photosynthetic bacteria which obtain their energy from organic or inorganic compounds in the environment. Alright, that's all about bacteria for now. We're still under prokaryotic organisms. Now moving to the archaea. So the archaea, they are also unicellular prokaryotic organisms. Okay, I forgot to mention that most, not most, but prokaryotes are unicellular. Alright. Okay, so the archaea are unicellular prokaryotic organisms. So its cell walls do not contain peptidoglycan, but they have a similar substance to peptidoglycan, which is called pseudopeptidoglycan. And sometimes you find that they said instead of saying peptidoglycan, they say murine. That's also find some books and literature refer to peptidoglycan as murine. And when it comes to archaea, instead of saying pseudopeptidoglycan, you say pseudomurine. Okay, so the archaea are found in nearly every habitat on Earth, and mostly on extreme environments that are very cold, very hot, very basic, or very acidic. Can you imagine that? So some of them live in the human body, but none have been found to be pathogenic. So in simple terms, you can say archaea are the microorganisms that can be referred to as extremophiles because they are adapted to live under extreme environments. Okay, now let's move to the eukaryotic microorganisms. These are those ones that fall under the domain eukarya, the largest domain of the three domains. Okay, so eukaryotic microorganisms, they can be unicellular or multicellular. Okay, remember protists mean unicellular from the proposed work that was done by Ernst Haeckel. Alright, so protists are unicellular eukaryotes that are not plants, animals, and fungi. So an example of a protist can be an algae or the protozoa. I know most of us have seen algae before. Okay, so the algae are like the greenish things that we can see on the waters. Usually when the water is left there unattended and it's flowing for quite a while, you'll see the green bodies building on that water. That's actually algae. Okay, and they are plant-like, and they are either unicellular or multicellular. So their cells are surrounded by cell walls made of cellulose, and they are photosynthetic. Okay, they extract the energy from the sun and release oxygen and carbohydrates into their environments. Okay, so one of the uses of this algae is making of the alga that's used in the medium that we use in the labs. Okay, that the agar is actually the gel derived from algae, and it can be mixed with various nutrients and be used to grow microorganisms in a petri dish. That agar you see in the lab is actually derived from algae. Okay, so they're also being developed as possible sources of biofuels. Another example of the protists are the protozoa, which are very diverse and they move with the help of the hair-like structures called cilia, or simply the whip-like structures called the flagella. Some protozoa are photosynthetic when others feed on organic material, and some are free living, and others can also be parasitic. So they can also be pathogenic and harmless. Now let's move to the fungi. So the fungi can be multicellular, such as the mushrooms, and they resemble plants. And fungi are not photosynthetic because their cell walls they are usually made out of chitin rather than cellulose. We also have unicellular fungi, which are the yeasts, and some yeasts are used for causing bread to rise or ferment beverages, and some can be pathogenic, such as the vaginal yeast infections, the oral thrush. Okay, so we also have the multicellular fungi, which are the molds. And molds play a critical role in the decomposition of dead plants and animals. They can also cause allergies and other diseases. And molds are used to make pharmaceuticals, including penicillin and cyclosporine, used to prevent the organ rejection following a transplant. Okay, so we get into detail about protists, fungi in more videos to come. This is just a brief video for microbiology. Now let's look at the helminths. So the helminths are multicellular parasitic worms. Okay, so they are technically macroscopic. We are able to see them without the microscope, but they fall within this field of microbiology because diseases caused by helminths they involve microscopic eggs and larvae. Usually the infection occurs after a person drinks water that's contaminated with water fleas or infected by the guinea worm larvae. Now the last or not the least are the viruses. So viruses are acellular. They are not made up of cells. Okay, and they should infect a living cell in order to reproduce. So we can define viruses as obligate intracellular parasites. Okay, obligate intracellular parasites. They should infect a cell in order to survive, and their infection leads to the production of more and more viruses, which will use the cell's ATP, and then after that they will cause the cell to lyse, then leave the cell and go and hunt for other cells to carry out another round of infection. Okay, and they also have cycles. We have lysogenic cycles and the lytic cycles. We'll discuss those later on the other videos. And bacteria, most viruses are pathogenic, but we also have viruses that infect the bacteria, which are called the bacteriophages. Okay, we have lysogenic phages and the T4 phages. Now before we conclude this video, let's go through some small exercise questions. You can pause the video and answer the questions. Then after that, you will see the solutions that I've put up for you here. So on our next video, we will focus on the properties of light, the different types of microscope, and the staining techniques. Thank you for watching.